Hi everyone, in this section we will talk about gene recombination. After watching this video, you should be able to tell types of genetic recombination for prokaryotic and eukaryotic cells. Gene recombination is also called genetic recombination. It involves the exchange of genetic material either between multiple chromosomes or between different regions of the same chromosome. Gene recombination occurs in both prokaryotic cells and eukaryotic cells. For prokaryote, there are generally 4 types of genetic recombination. They are transformation, transduction, conjugation, and protoplast fusion. Firstly, let's take a look at transformation. Please look at this picture. It briefly shows the process of transformation. The recipient cell absorbs the DNA fragment from the donor. Consequently, they acquired some traits from the donor. Whether the transformation can occur between 2 strains depends on their phylogenetic relationship. However, it is not necessarily the transformation could occur even among the microorganisms with high transformation frequency. It is found that recipient cell must be at the competence state, wherein it is easy to accept donor DNA. The second type of genetic recombination for prokaryote is transduction. Please look at this picture. It shows the process of transferring of genes from one cell to another by bacteriophage. In 1952, the Nobel Prize winner G. Ledberg discovered this phenomenon when he studied the recombination of Salmonella typhimurium. Transduction can be classified into generalized transduction, specialized transduction, and lysogenic conversion. The third type of genetic recombination for prokaryote is conjugation, or called mating. In 1952, microbial genetics W. Hayes and Ledberg proved that E. coli cells also have male and female types, which is related to the presence of a plasmid called F factor, also known as sex factor or fertility factor. Bacteria with F factor are donor bacteria, while bacteria without F factor are receptor bacteria. Please look at this picture. When the bacteria are conjugated, the donor cells are connected to recipient cells through sexual umbrella hair on the cell surface. At the same time, the donor's chromosome DNA is transferred from a single strand to recipient cells and recombined with recipient's chromosome. The fourth type of genetic recombination for prokaryote is protoplast fusion, which refers to the fusion process of protoplast of two cells with different traits to obtain stable recombinants with both parents' traits. The fusion between two protoplasts is realized by artificial method. It was developed in 1970s and often used as a breeding approach. There are many kinds of organisms capable of protoplast fusion, including not only prokaryotes but also eukaryotes. On the next, let's take a look at the gene recombination for prokaryotes. There are many types of gene recombination for eukaryotes, such as sexual hybridization, parasexual hybridization, protofusion, and gene genetic transformation. The latter two are similar with that for prokaryote. Herein we will focus on sexual hybridization and parasexual hybridization. Generally, sexual hybridization referred to sexual reproduction of hybrid organisms by hybridization of two different genotypes. In principle, any yeast, mold, and mushroom that can produce sexual spores can be bred by sexual hybridization. Please look at this picture. It shows the morphology of S. cerevisiae, a species that can be used in wine making. Here we will take S. cerevisiae as an example to briefly introduce the process of sexual hybridization. Parent strains with different production characteristics suggested as strain A and strain B in this picture were inoculated on the medium containing sodium acetate to produce ascars. The haploid cells from different parents were closely contacted by centrifugation to produce various diploid sexual hybrids. Hybrids with good traits can be screened out from diploid hybrid. On the next, let's take a look at parasexual hybridization. It is similar to, but more primitive form of sexual reproduction. This is a fusion between the somatic cells of different strains within the same species. It can cause low-frequency gene recombination and produce recombinants without resulting to meiosis. So, parasexual hybridization can be regarded as a spontaneous protoplast fusion between cells of eukaryotic organisms. It is a common phenomenon in some strains of fungi imperfecta. Finally, let's take a look at the keyword in this section. Okay, that's all for this section.